All right, we're going to get started here. Thank you, everybody, for coming to our workshop. It's my pleasure to moderate this panel. My name is Andres Castillo. I'm a cell and gene process manager at Sartorius, an innovative leader in and supplier of biotechnology products. And I'm very honored to moderate this panel. I know there's a wealth of experience, and I anticipate a very lively discussion. So without further ado, could you please introduce yourselves and what you are excited about in the cell and gene therapy space in the next 5 years?
Dave Smith, President and Chief Commercial Officer at Akron Biotech. Before this, I was with Helios in Japan, and 16 years before that, I was the employee one and built the Lonza cell and gene therapy business. So a bit of background on that. What I'm excited about is I think that moving forward, the jump from liquid tumors to treatment of solid cancers is going to be a step change. But it's going to force a lot of manufacturing changes to go with it. We aren't going to be able to have autologous to meet those kind of demands. We're going to need a lower cost, high yielding process to treat a large number of patients.
Hi, Jessica Carmen. I'm Business Development Advisor to Oxford Biomedica, which is a viral vector manufacturer based in Oxford, UK, with now a joint venture recently formed with Homology Medicines called Oxford Biomedica Solutions in Boston for the manufacture of AAV. I would say that what I'm most excited about is that I'm really excited to see how many gene therapy and gene-modified cell therapies have been approved in the last year and a half, and looking at how many of these products are on the docket to be approved in the next year and a half or more. It's making you realize that the need for commercially manufactured viral vectors is quite large, and the ability to manufacture large volumes of viral vectors is ever present. And the technology is there and ready and able to meet that need. And that's going to be exciting to see all these products become available to patients.
Absolutely. Jason?
Jason Foster. I'm the CEO of Ori Biotech. Relatively new to cell and gene therapy in the last few years, but 25 years in biopharma. I think the thing that's most exciting for me is the opportunity to achieve widespread patient access to these therapies. I mean, we have incredible therapeutic efficacy. We've seen recent 10-year data coming out from some of the very first CAR T patients offering the ability to say we've cured patients with cancer, but only very, very few, unfortunately. So really that's the big ambition in the next 5 years. We'd love to see widespread adoption and treatment of patients at scale.
That's incredibly exciting, and I think patient access has been a big theme of the meeting, so yeah, definitely excited about that. Andy?
Sure. Andy Holt. I'm the Chief Commercial Officer for Incardia. Spent a good bit of time, it's 12, 14 years in the cell and gene therapy space, really focused on manufacturing and testing, kind of the scale up and quality control functions. Right before I came to Incardia, I spent about 4 years at AskBio, a gene therapy developer in RTP, based around some really fun AAV technologies that I was lucky enough to play with for a while, and excited for kind of applying that same sort of mentality to iPSCs. I think for me, there's a real gap in that'll be closed one way or the other in the next 5 years between the science of these therapeutics, which is humbling and impressive, and then the engineering task of actually making them in a way that is controllable and repeatable, and it addresses a lot of the stuff that my other colleagues here on the stage have said about accessibility. I mean, I think right now it's kind of a tragedy that if you're in the right place or born with the right economics with a rare disease, you have a much better potential outcome right now than if you do if you happen to be born in a different place or in a different economic situation. And it's the task of manufacturing and scale up that has to be focused on, and we're lucky enough to inherit these tasks based on kind of shoulders of giants problems, right, from science that's been baking for 20 or 30 years. So that's humbling and exciting to have a role in that.
Yeah, no, absolutely. And I think, more and more partners are trying to sort of bridge that gap, right, between the complexity of manufacture and building that accessibility. Francois?
Hi everyone, I'm Francois Generim, Senior Director, CMC Quality at Voisin Consulting. Company exists for 25 years. We've accompanied a number of projects. I think it's fair to say ATMP development has been in the DNA of the company for even before that was an actual product category, which is not so long ago. And happy to be here with everyone, and not to repeat what was already said on how we can capitalize on the last 10 years of development. What I am looking forward is, everyone is on the same boat on this. If one company fails with a product category, it will be a step back for the other because they may have overlooked something that ended up being a safety issue. So taking the opportunity of those meetings, the forums that the ARM offers, other opportunities to see how we can align. I think there is really room for everyone considering the number of diseases we have to treat, and see how we can help each other. And I think one way to do that is the regulatory framework for this product is 15 years old. It will be revised soon. The compendial referentials are going to evolve as well. There will be standards for this. There will be standards for methods, raw materials, substances that everyone uses at the moment, and we all know that the quality grade is not what it's supposed to be, and how do we qualify this? And it's a common effort, I don't think one company can do it alone, because it would be too much of an investment to get to a point where this becomes a standard. So I'm really looking forward to this and see how regulatory perspectives and manufacturing constraints align so that we can give access to those products to more patients than what we have done for the last 10 years.
Yeah, absolutely. And I kind of want to keep on that theme. So what bottlenecks do you see in production, and just staring at the problem in the face? What keeps you up at night, right? And we can go down and share our thoughts on that. Yeah.
What keeps me up at night is it's going to be a pivotal phase. Everyone is complaining about the lack of regulation and standard on some aspects. Everyone wants a checklist, and everyone will cry once the checklist is there, because then you have to follow the checklist. So what keeps me up at night is making sure that everyone can be pragmatic about it, not going too much into one direction that may not be the standard for tomorrow, keeping the flexibility so that innovation can continue to thrive, and at the same time facing the global responsibility to move from what used to be quite handmade product, even the first ATMPs that have been approved, both Europe and US. If you look at the process, if you look at the level of flexibility the authorities gave in their assessment, it's crazy. It was a world of opportunity. We needed those products. You have to start somewhere, but we should be learning from this. You almost have subcategories of product right now. As a CAR-T is a category of its own. You have specific constraints. Some tissues are very specific as well. So, yeah, making sure we improve quality, looking for efficacy, because that's the downside. If you don't have it, then there is no product. But still remaining pragmatic and not going too far in having standards that then no one will meet anymore. And right. I think that's the point for me, yeah.
Yeah. Andy, do you have anything to add in terms of what keeps you up at night?
Yeah, it's strange the moment kind of we're in right now, and I will gift one of my other panelists the COVID answer. So one of you can take it. I will try to do something else. A couple of years ago, it was waiting on a trial readout, right? Or hearing that there was an adverse event in a particular trial that was prominent, and looking at the impact it would have on the industry, and worrying that the projects that I was involved in and were passionate about would be impacted by somebody else's choices that were not my choices, and that there was a lack of understanding of the modality and the fundamentals that would kind of splash that damage around. I know, Francois mentioned a bit of this. I think we're moving past that right now, honestly, which I'm grateful for. I think now I'm more worried about supply chain than anything else, that circumstance has a couple different aspects to me. One is that for some of the things that are most scientifically and successful and scalable, they're not of appropriate quality, and so we're just kind of waiting for somebody to figure that out. I think that's a supply chain challenge we've given ourselves by being in perhaps a justifiable hurry to get these therapeutics moving. It's hard not to be excited when you have revolutionary results to kind of do good enough and not be ready for what success looks like long term. And I think we're all, for better or worse, relying on the same like 8 or 9 things across every modality in gene and cell therapy. And as this industry kind of explodes, I think there's a real obligation on the suppliers and service providers to take a little more risk, to get a little more. If they build it, they'll come and deal with the possible downsides of that, than just say, well, we'd like to, but it takes 18 months for capacity. It's like, well, you knew this was coming. And patients are suffering because you were unwilling to take the risk. And I think we can, on this side of the fence, so to speak, change our risk appetites and help, to belabor the metaphor, people sleep at night.
Yeah. Jason, do you have a follow-up?
At the risk of sounding potentially too generic, through the last couple of days, as you drill down into some of the challenges that we face as an industry, you can get kind of very specific and nuanced. If you try and chunk it up a bit, I think one of the things that should be keeping us up at night is trying to apply old models to a new modality. So we're thinking about manufacturing in the way we've been manufacturing biologics and small molecules for the last 20 years for fundamentally different products and living medicines. And is that the right framework to bring forward? Many of us, I mean, I myself came from small molecules, many of us came from biologics or other fields, and we're bringing that baggage and experience with us. Some of it's good, but some of it might not be as helpful as, we need to revisit some of those assumptions and the frameworks that we're using to think about this new modality and apply different thinking to it. So just as an example, manufacturing or distribution, you think that the ultimate failure, in my view, is going through all the pain and suffering of developing products that are clinically efficacious, that are then approved, but then can't be delivered to patients at scale. So you might get a couple patients that benefit, or even worse, you see a product which will go unnamed, be withdrawn from Europe when it is incredibly clinically effective, but just can't be afforded or can't be available for whatever reasons. And that is the ultimate disaster. Disaster for the industry, disaster for the developer who spent billions of dollars in 10 years making it, disaster absolutely for patients. That ultimately is the worst case scenario and should be what's keeping us all up at night, is that we can see that happening over and over again potentially to the next round of therapies, the ones that have been approved in the last year, the ones that are about to be approved in the next year or two. So what do you do about it, Jason? It's great to highlight the problem, but let's come to it with a solution. We say one of the interesting pieces is we need to not make the same assumptions throughout the process. So it used to be if you got good clinical data, someone would come buy you out and it'd be someone else's problem, right? Manufacturing, someone else downstream, you can worry about that. All I need is good phase 2 data and I'm golden. Big pharma is going to take me out. That's not the case anymore, and that's not the case in cell and gene therapy. That ship has sailed. So how do we need to think about it differently as investors, as innovators, to say, is it worth taking the time, rather than rushing to the clinic and getting that clinical stage company badge and raising our $100 million, at least when we could fundraise a year ago, is all of that the right process for us now? If we reframe success as how many patients have we successfully treated with a therapy? If that is the ultimate end game, would we do things differently along the path to try and reach that goal? And so maybe that's something we can explore in more detail. I'm interested in the audience's view and the other panelists' view. There is an old model that has been successful in the past. Is that the right model as we move forward?
Right, and I think that's the sort of the foundation of this panel is understanding where in the process can we learn more and take those learnings and scale those learnings and build a robust process development or early discovery pipeline to then support that.
Yeah.
Jessica, do you have anything to add?
Yeah, so I sort of piggybacking onto your comment, I think clinically effective, non-commercially viable really is, just a horrible place to be for any product. It's horrible for developers, it's investors, patients particularly.
Everybody.
And I think there's so many directions you could go just on that right there. Is that due to a lack of technology? Is that due to a lack of patients to be able to adequately test to see if the clinical tests are not really, I mean, there's so many directions we could go with that. I'll choose to go with the technical route, which is I think sometimes there's a lack of robustness in early phase products. And sort of what you were saying, let's just push this through quickly and pharma's gonna pick it up. Well, pharma isn't like the magic angel that's just gonna, bless it, and there's work that has to be done. There's either, process characters, actually there's definitely process characterization that has to occur. There's gonna be technologies that have to be applied. There's scale up that has to be done. That's a lot of work. And little to none of that often does get done early. And I think that's really what the biggest problem with this bridge of, from efficacy to commercial success.
Yeah, 100%.
Yeah, I was going to go along with the team. For us, we've been talking about it since the early years of cell therapy. People don't characterize their cells often enough, early enough, because what always happens is the same thing. We get a process in phase 1, it looks great. You might meet some regulatory standards in a country or two, but then what happens is you get to the next step and once you have great results, you have medias that have small molecules in it that you can't get GMP or never will get GMP. You need to scale up from a small scale into a large scale. I have to move from a cell factory into a bioreactor. No one knows what these cells are going to do when we move to that next step. We've talked about this as the problem for 20 years, that we don't have good characterizations, we don't have good potency assays, we don't have good efficacy, purity assays. And it's funny, we come back to the same thing. We get processes from all over, from academics and from small companies, and everyone wants to just kick it down the road. Hey, you know what? I'm going to sell it after phase 1, 2. It's pharma's problem after that, and then you can't scale it up. And so pharma has become much more aware and much more engaged in this. They know this is a problem. Companies who come to them with great potency assays get a priority, and the manufacturing groups just love you. I mean, because in the end, I want to be able to get your costs down. I have to show that I can take you from 50 patients to 5,000 patients and make the same cells every time. How do I do that? I have to be able to scale it up, and I have to make changes. It's even more, in autologous, it's somewhat more simple, but when you go to allogeneic, again, we start talking about this shift from going from blood tumors over to solid tumors. Right. How do you treat 20,000 lung cancers? Because this becomes a primary, it goes from tertiary all the way up to second-line therapy, primary therapy. Now you're talking a lot of patients. How do we do it? It comes down to what did you do at going into your IND, and what did you do in phase 1? Do you have a product that can be scaled? And in the end, that's the solution to everything. You characterize, know your cells.
Right. But I think, the incentives there are misaligned, right?
Yeah, I've said that a few times, haven't I?
Yeah, but I just think that's a really interesting, I've only worked with a few groups who had the luxury of resources or time in preclinical to actually do that. Because it's not just like I don't hear big pharma with an angel noise that often in my experience, but maybe they aren't. But whether if we're being uncharitable, it's the value creation and acquisition so everybody buys a boat, right? If we're being charitable and kind of maybe the world I want to live in more, it's you're getting to patients. And fundamentally, right, in these therapeutics, you don't know if they work until they go in a human being. Because mice lie and monkeys lie and cells lie and everybody lies to you throughout the whole development process. So you're going on moderate safety and minimal efficacy and hoping. But again, just the nature of the industry, right? We're dominated by science-heavy dreamer people who've had this is their thing they want to do for 15 years, and they know all the patients and they know all the families. you're not incentivized. You're incentivized to know if you actually have something by getting it into people, because there's not a good way to know until you get into people.
Right.
And frankly, our industry has had serious adverse events from that phenomenon, right? But you're all of your incentives are to get to that point, because that's the first time you have real data, so your value inflection happens too. But nobody cares. Like, nobody cares. And so you have to go out on this lonely little island, like maybe it's a peninsula if you're lucky,
Yeah.
so there's a way back off of it, where you put all this time into process development and characterization, do all the stuff you said. Nobody pays you for it if your molecule fails.
Yeah.
Then you have an empty house that you overbuilt.
Right.
And I don't have, I'll do the opposite of what Jason, I'll just complain with no solutions.
We'll join you in a minute.
That's
Yeah,
fine, but
complain.
you see what I'm saying? Like, there's a fundamental misalignment of incentives where nobody would actually want to do that. I want people to do that real bad, but if you're talking to boards and investors, why would they bother?
So here's a really good question there is, we've seen a proliferation of companies that are platform-based, right? I mean, this is kind of the new thing, right? And so in theory, shouldn't there be the ability to characterize and learn a lot about the platform? And then as you peel products off, there's a lot of learning from the previous product. So if that's been happening, why are we in this situation still?
Yeah,
Well,
but
it's not
I come
actually
back,
happening.
Andy, I come back to who's the
It's just in the pitch
poster
deck. It
child
actually
for
happened
not
in reality.
not characterized, not good validation. Our friends at the till, there's a certain company who has a till program that how many times, I mean, we know that process works. It's been run for 25 years, and it got stopped at the goal line because of an assay. I mean, that is what we should all remember. Francois wants to.
Now, I'll try to go on the options that we have. There is one incentive, and it's not a good one, but there is one.
Well, bad incentives.
Yeah, a bad incentive. No, but I mean, everyone is asking, okay, why is it faster to get into first-in-human in US? Potential assay is one of the major components of the answers, is because the FDA said, you know what, we are okay with first-in-human with one single potential assay. It can be a surrogate. You will do correlation later. European authorities said, you know what, we want you to explore multiple potential assay candidates because you can't know at the time of your first-in-human that this is how you measure your quality parameter properly. And if you have an issue later and something you need to explain, you won't have the comparability point in time to justify this. So they don't ask you to have multiple potential assays with multiple specifications. They ask you to have multiple candidates. You still select one surrogate, but you have something to compare with. That's a bad incentive because it creates differences. Everyone goes for first-in-human in the US, and we still have the problem. To be fair, I think the FDA is trying to change this. If you look at the question you receive on first-in-human right now, it's not exactly as freelance as it was 5 years ago. It starts to get more complicated than just, viability plus identity plus maybe one thing that looks like it's functional enough, it's sufficient. They start to go the same way. And I think the European authority is trying to be slightly more pragmatic about it because it's at the national level, so you can't hope for harmonization. That may be another topic that keeps us awake. But they are trying to be more pragmatic about it because they want to catch up. They don't want the product that arrive in Europe for the pivotal phase with no first-in-human. So that's the idea. Second option is there are examples of companies. It was not a good news at first, but managed to develop a potential assay that was good enough so that they would sell the potential assay to other developers before they had an actual product. It has happened for CAR-T. They had a good cell-killing assay, fully functional, easy to validate. They sold it to larger companies. They became an analytical supplier before they became a drug product company. So I'm not saying that's a story for everyone. I'm not saying that's the example to follow. But that's one solution. That's something that does exist. And at the end of the day, it is how you show, and I get it. You are in the room early in development with the investors. You have the developer on one end. You have the guys on the other hand. The question is sometimes, do we pull the plug right now, or do we wait for another 6 months? When you're in that context, selling them the idea, you know what, we need another year to explore some aspects of our mechanism of action that we will never fully understand, but this is an additional brick to the walls. That's very important. So solution is to explain what is the value. And even if you have an exit strategy after a first clinical trial, hoping it is successful, I think there is education on showing that, you know what, we've explored that mechanism of action through characterization. It has value. You don't have to do that again. And maybe if you have a hard time validating the process because the process is immature, you still have some data on the quality parameters that will support moving from that initial process to the commercial one. I'm not saying it's easy, but if we wait for incentives to spend more money early on, but while maintaining the uncertainty on the potential of the product, I'm not sure that will happen. If we explain that this is money well spent because it removes the risk later on, that's an angle. I'm not saying we will be successful everywhere. That's an angle.
I mean, I think for me, a good potency assay doesn't fix the problem if you've got a process that's too slow, too expensive, it's going to deliver a high cost of goods product that's low throughput and low quality. You haven't fixed any of the problems. You got a good assay, but you can release the product, but you just can't treat patients. Because I'm not a scientist, all these people are much more qualified than I to talk about this issue. I'm a simple person, and this is a supply and demand issue. It's, can we find the right economic incentives to enforce the behaviors we want to see earlier in the process? Why did 5 or 6 or 7 companies jump into COVID-19 vaccines immediately? Because they knew they were going to be reimbursed. They were 100% going to be paid for. There was a giant market opportunity to go after, and so they ran after it hard. The regulators played along. They would flex the rules a little bit because we needed to. That kind of model is not a million miles away from where we might want to explore that space in cell and gene. These are incredibly sick patients. Many of them, this is their last treatment option. What's the harm in relaxing some of the structures, the controls around this to align incentives, to take the time to create a process that could treat more than 1% or 2% of the patients that it could clinically help? Right. And I think part of the answer is what Andy was saying, is to say, potentially the financial incentives are realigning themselves, right? If there isn't that phase 2 exit, this is going to be someone else's problem, then how do investors make their returns? How do companies, create value if it is by reaching the market at scale, whatever that means, versus good phase 2 data or approval, which is, when you think about how hard you fight to get an approval, and you've just gotten to the starting line of trying to treat patients and all the things that come after that. Because I'm a small molecule guy, I think about things like market access. We don't talk much about market access in cell and gene therapy because we don't have to worry about that right now because we're struggling so hard to get products to the market. But this is something that's a real challenge, and it's going to continue to be a real challenge. As more products get approved, it's going to get harder and harder to find room in those budgets to pay for these products. It used to be, if it works clinically, it'll get paid for, at least in the US, and we'll worry about Europe later. That was kind of the attitude. That attitude doesn't work anymore. And so thinking about how we provide the right incentives in the system to people to take the time to develop a process that works, that's scalable, that's repeatable, that you can validate.
Right. And sorry, I was.
No, that's it.
No, we're all good now.
We can go off on this.
Yeah, I know. It was just such a great discussion, and I think I just want to, maybe looking at the process, maybe it's building that process in early, right? So connecting the process to the cell characterization early in development and really building out that platform. And I just want to, naturally though, connecting, the upstream, the downstream, and the data analytics within it, it creates these natural silos of data, right? And so I was wondering, do you have any opinions on, managing those silos and connecting those and building those out? Data.
And you hit it on the head. Part of it is understanding what you don't know on a lot of this, because a lot of it is going to people who screwed up. I mean, let's be honest. We all learn by this, all the screw ups we've lived through.
Yeah.
I take a really simple one, and Francois, this is going to aim at you, which is right now everyone still has some protein in their whether it's HSA, HPL, AB, very simple. What am I going to use? Well, the regs for Europe, the regs for Japan, the regs for the US, completely different. And different testing, different requirements. Right. So you start with the basics. Okay, I find people who know what they do. I break down my process into individual components, whether it's raw materials, the actual process steps, the assays, and ask everyone.
Right.
That sounds silly, but if you do the work early and figure out how to put the data together, have your assays lined up, at least some qualified assays, it's amazing how much smoother it's going to go going forward, and how much people will value it and put value behind it as you step forward.
Yeah.
It's interesting to realize how many of us up here have worked for CDMOs, the same one, actually. But in reality, CDMOs are a treasure trove of this information, because rather than going to a developer who has data on their process,
120?
yes, the CDMOs have all the data on all these, and there must be some way that the data could be sort of anonymized, scrubbed, whatever, just to actually look at process,
This
parameters.
is my favorite point. This is my, if I have anything in the world, this is my favorite topic, and I'm sorry, because I hope you can relate. But the problem is that, again, when you talk about misaligned incentives and the framework stuff, the CDMOs don't own the processes they make, because they're not incentivized to own them, so they don't own the data. So all of us I've worked for a bunch of CDMOs, and I'm here on stage representing one we announced yesterday or the day before, whatever, right? Owning the data is really important, but CDMOs can't do that if they don't own the process, so they have to invest in the process themselves, or else they're going to be doing nothing but tech transfers. And then, I will tell you, I have developed an adherent AAV process in HyperStacks 14 times, right? And you have to develop each step of it, and it all looks exactly the same with the same medias and the same stuff, but you have to develop it again. And you can't tell, I've been on plenty of project teams where you watch somebody go, well, I want to do this thing. It's like, that's a dumb thing. And it's going to screw up. And then
advantage.
Because I can't tell you, because the data is confidential, that I've seen that mistake happen 5 times. Right. So that's when my last company and this company were trying, at least, right?
Yeah.
And it was really fun in my last role, where we'd run a single AAV process 745 times right before I left. And so we had Six Sigma, real data-driven. We had a data scientist, and all she did was run validation reports on Excel, but it's because we owned the data. And without that but we had a process that was developed with our partner company, so we were okay. CDMOs, unless you're willing to jump out on that ledge and say, this is the way we're going to make it, and then have every company that comes to you take pot shots at the way you make it.
So that's what Oxford Biomedica does.
Right.
Actually, so Oxford Biomedica has a platform technology for lentiviral vector manufacture, and everybody that comes in, we do have the option to tech transfer in, and we'll just make your process however you give it to us. We'll run your recipe. But we ideally and encourage people to use the platform we have, because we do have all of that data on the platform. And in fact, in 2019, I think it was, we announced a partnership with Microsoft to help us sort of manage and go through that data to be able to make sense and learn from the platform.
Right, and it's so much better, but then, you have to take that assertive step first.
Don't always love the idea of using somebody else's process.
Jessica,
but
how difficult is it? We had self-factory process, lockdown, top to bottom, closed system. The number of customers we said to, we have a process, we have a batch record, please just use this. And they said, nope.
Because manufacturing IP is valuable for your...
It's about manufacturing IP.
Indeed. Even if it's unsupported IP that's never been used. It's a weird phenomenon.
Trade secret.
Yeah, so it seems like it creates a real incentive to sort of build that process out, understand it, and then be able to communicate that value, internally, externally as well. The next question I have is, in terms of adopting new technologies into the pipeline, how do you assess or communicate long-term value within that? And how do you sort of, encourage this new way of doing things, within these processes?
You want to give me
Jason.
that one? I have a feeling you might throw that one to me.
Yeah.
Well, it's very interesting. I get told I'm crazy a lot. On the data piece, just to kind of, one of the core pillars of the Ori strategy is to have a cloud-native IoT device that can capture data, multiple sites. You can aggregate it, you can structure it, you can analyze it, and you can turn it into information. And so that could potentially be useful to drug developers, help shorten drug development times. But everyone says, well, pharma's not going to give you their data. And I say, well, they'll give it to me if it's valuable enough for them to do so. And it's up to us to be able to demonstrate that. But adoption cycles are long. Biopharma is risk averse. We all know this. I think, luckily for Ori, we're a bespoke manufacturing platform that was built for cell therapy. So we're really trying hard to solve the problems, some of the things we're talking about today, and not repurposing other square pegs to try and to jam into round holes. But you need reference customers. No one's going to say, I want to do it first. Who else is doing this? Who else is adopted? It's all those sort of normal tech adoption kind of things that you would expect. But it really is about value. It's about, what we see as part of our role is to take some of the heavy lifting off of therapy developers. Right. What are therapy developers good at? They're good at finding new modalities, deploying them clinically, delivering clinical outcomes for patients, running clinical trials, sales and marketing. Those are the things they do great. They've had to develop manufacturing capability because someone had to, and they had to get products made. Right. Is that the best solution to go back to my very first point, is that the best solution for cell and gene therapy, that everybody owns their own manufacturing capacity? You could argue potentially it's not. There are other ways to deliver these modalities that rely on technology that would be less expensive, higher quality, higher throughput, more consistent, maybe shortening vein-to-vein times, improving clinical outcomes. I can say all the buzzwords to say that that's what needs to happen. And it's, how do you get over the hump? You have to demonstrate it one time, and then you have an opportunity to demonstrate it again. Right. And what we're looking at is how do we do, because it's a cloud-native platform, you have the ability to do the same process in two different places, monitored centrally, demonstrating you get the same output using the same inputs, which is all really anyone wants to see, right? That's what the regulator wants. So it takes time, and it takes being sure that we're trying to solve for what therapy developers or CDMOs or whoever that partner is wants. And that use case, that value proposition is a little bit different for each of those audiences as it relates to technology.
So going off of that, so what about scaled down models and having really efficacious scaled down models early in development? Can someone
First, you have to scale up.
And then you scale back down.
Usually the other way, but,
that's true. But creating like sort of this effective platform early on to then scale up into production or screen multiple parameters and things like that.
So what I've seen so far is, especially when you go to the bioreactor side, it very quickly became kind of the rest of bio mindset. If I work at 10 liters, I can go to 100 liters and people kind of and it's pretty accepted that that's a nice passage, especially since you have a lot of intermediate assays you can do. You're getting a lot of feedback off the tank. You're getting that, and it's pretty acceptable. Off the more manual scale-ups, it's not as prevalent.
Suspension is scalable on any modality, cells, viral vectors, proteins.
So it's going back to, it's almost once you get away from, if we get away from straight autologous, where it's more about equipment and throughput, we go more towards the cell therapy scale up that we're going to. It's really starting to get much closer to bio as we expect it.
I agree. I mean, I think historically the focus has been on the technology, whereas potentially focusing on the broader solution set. It could be multiple technologies that are pre-integrated. It could be pieces from different players that do those pieces well, but trying to solve the problem holistically. And again, taking the burden off. I mean, trying to combine a bunch of technologies that weren't intended to work together. That's just a giant pain in the ass that the industry doesn't need. So us, we put the monkey on the back of therapy developers and investors earlier, taking it on our own shoulders to say, as technology companies, as enabling technology providers, it's up to us to work hard together in trying to integrate these solutions, and talking to people like Oxford Biomedica, for example, on their viral vector production. How does that fit into the system that we're building? And there's all these pieces. And historically, maybe tech providers haven't played nicely in the sandbox together. But there's an opportunity here. I don't want to do what some of these other vendors do, and I'm not a CDMO, but I can work with all of them to make the solution more comprehensive and better. Right. And that's how a lot of the folks on the stage that I've met them is through those partnership conversations to say, how do we take your solution, integrate it with ours, and make a 1 + 1 = 3 scenario? And there's a lot more of that to be done, in my view.
Yeah, no, I think definitely there's a lot of partnership, internal knowledge, just sharing it. I think that's been mentioned before too. In terms of, you know, I think it's this complexity, right? The cell is inherently complex, and the environment in which the cell grows obviously, you know, is directly correlated to the efficacy of the final cell product. How do we go about sort of controlling the inputs, right? In autologous therapy, you have multiple donors. How do we start with that characterization? Francois, you have.
I mean, there is inherent variability to how those therapies work. And the responsibility is to remove all the variability that you can remove. Starting material, if it's autologous and even if it's allogenic, you will have a hard time removing this. Switching to iPSCs might be one solution. There are other type of cell lines, but that's one of the high hopes, is that when you get the well-established cell line and your process is robust enough so that you always obtain the same differentiation, you get rid of the starting material-related variability. That's one of the options. For autologous, the impact of variability of the starting material is not as important if you consider the clinical use, because it's what you have, it's the patient you have. At the end of the day, especially when you look at what those products are used for, it doesn't matter, you know. You wouldn't believe how many of those batches are really out of specification anyway, because the specifications have been designed for a purpose that is not exactly aligned to the chances of the patient. And if it's the only chance they have to receiving something, you won't deny them a treatment because you're out of spec. So to me, it's a separated approach, but it's not scalable, because if you're autologous, you're autologous. So opportunities to remove variabilities. Improvement in the quality grade of the raw materials that are used. And I say that completely understanding that in cell therapy, it's not straightforward. You use a R&D medium, and the cells are very happy with it, and you switch to the GMP one. And GMP raw material is something that could be a topic for an hour, but let's say the clinical grade, which should be the definition.
See Dave Smith after
You switch to clinical grade raw material, and suddenly the cell don't grow the same way. You need to add 2 passages, and because you've added 2 passages, you start to see something that looks like a beginning of unstable genetics. It creates a lot of problem, and the cleanest material is not necessarily the best one. So we all say, oh, you know what, you just have to upgrade. It's not that easy. Sometimes there are materials you can't replace because then your process no longer work. And back to what we were saying, if you don't understand why, you have no solution. Most of the time, you are still able to carry on with that. And I think the risk then is, what happens if someone comes with the same product, same indication, but they figure that out? Then your product is, and we go back to the market access, but the value that is behind it. So you would have maintained something that was substandard in a way for long enough. You manage. Everyone is happy because you're on the market, and the product that comes out 2 years later, they don't have that issue. So that's a very difficult discussion to know where you can apply pressure to reduce variability, because it's definitely not straightforward in terms of science, and it's not straightforward in terms of regulatory as well, because we were talking about how to implement innovation. This product category is mature enough that innovation is no longer considered necessarily as a good thing. You know what? 10 years ago, all excipients that you would be using injected in humans, they will all be novel excipients, never been used in that route of administration for such product. Nowadays, maybe some people have a very innovative formulation, and everyone is afraid to use it because you don't want the extra burden of doing that work on the top of everything else you have to do. Same for the closed and automated system production, you know. It's basically like there are only a few companies that are willing to be the first with those innovations, so we are depriving ourselves with the opportunity to implement that faster, just because even if it's not a standard yet, you will be evaluated against what has been approved already. Mm-hmm. So.
Francois, I got to throw one in here. Number one thing that we found all through it, so many of the things you work with are based off of international units, and so take every one of your raw materials, knock out the variability you can. If you do lot trending on different lots of raw materials you're getting, you're going to find a very big swing on these things. And as you start, I mean, it sounds really simple, sounds really straightforward, but you can see a 30 to 40% swing in raw materials, and you're wondering why a patient doesn't grow. Well, it might not have been a bad patient, plus you had a low international units.
Yeah. Well, it's the context of the data, right? And a lot of what we've been talking around, like to me, kind of boils down to that of the therapy has variability, but unless you have a platform, right? Unless you can really generate the data that you need to put the variability in context, you don't know. So you can't adopt new technologies. You can't do downscale because you're opening a black box because you didn't do the work to start.
Yeah.
And that's where I think like so much of this for me boils down to a lot of companies move to internalize manufacturing in these modalities, which is the exception rather than the rule. When you look at other modalities, like there's very few, it's just unusual. They had to because we on the manufacturing and partnership side ran out of capacity, didn't take the risk, weren't good partners. They internalized it. In internalizing, they lose access to data, and now they can't answer the questions. And that's what, Jason mentioned it, taking some of that back is what, a lot of us are all about here, right? I want I want to have a platform for iPSCs so you don't have to care about differentiation. We will be able to contextualize that data.
Right.
Same for other cell therapy, like for your tools, for
Yeah.
I could do that.
And you always,
I know.
as always,
I
the last
feel like
word.
we could.
yeah.
No, thank you so much. And I think everything that has been said is so relevant. Controlling that variability, leveraging partnerships, and building robust platforms. And so I know we're the last thing stopping you from the last network event, but thank you so much for joining us. Thank you so much for joining me on this panel. It's been a wonderful opportunity to hear all your insights. So thank you so much.
Thank
Thanks
you.
for having us. Thanks.
Been fun.
Thank you.